subject I'm going to talk about today is attachment and replication of PRRS virus on bone marrow-derived dendritic cells. This study is mainly focused on the receptors that the PRRS virus used for the infection. So, firstly, let's have a brief review about the 6 receptors that have been found. Heparin sulfate, the initial contact of the virus on the cells, and sialodohexane, CD163. The 2 main receptors, especially the CD163, is generally considered as the essential receptor for PRRS virus infection. Except this, there are also other receptors like vimentin CD151, and also the DC-SAM, which is highly expressed on dendritic cells. Now let's have a deep looking on the 2 main receptors. Sialodohexane interacts with the matrix protein and glycoprotein 5 to mediate the viral attachment and to some extent the internalization of the virus. CD163 interacts with the trimers GP2A, GP3, and GP4, leading to the virus internalization and genome releasing. However, recent studies indicate that other receptors may exist for the PRRS virus infection. For example, the highly virulent isolate LINA was reported to infect CD163 and sialodohexane negative cells. But most studies on receptors that have been done were on alveolar macrophages. Alveolar macrophages are a homogeneous population in the expression of sialodohexane and CD163. It is not the same case for the dendritic cells, which also could be infected by the PRRS virus in vitro. Dendritic cells are a heterogeneous population in terms of this cell surface molecule expression, and the PRRS virus was supposed to escape the immune response by infection of dendritic cells. The objective of my study, 1, is to examine the attachment of the 3 genotype 1 PRRS isolates to bone marrow-derived dendritic cells, and the relationship with the 2 receptors, sialodohexane and CD163, and also the relationship with activation status of dendritic cells. Another is to examine the replication of these 3 isolates in bone marrow-derived dendritic cells. The 3 isolates, 3249, 3262, 3267, was well characterized by our group before. Methods. Bone marrow dendritic cells were obtained by treating bone marrow hematopoietic cells with recombinant Porcine GM-CSF in vitro for 8 days. We defined these cells as immature bone marrow-derived dendritic cells, and after overnight treatment with LPS, we got the mature ones. Then we used the 2 cell types for viral attachment and viral replication next. In the part of viral attachment, we incubated immature or mature bone marrow-derived dendritic cells with the 3 isolates separately, with the multiplicity of infection 1 at 4 degrees for 60 minutes. And then we fixed the cells and did the immunofluorescence staining for the virus and the receptors CD169 or CD163. In the viral replication, the multiplicity of infection was changed to 0.1, and we collect the cells and the supernatants at 4 different time points, 0, 12, 24, and 48 hours. For the cells, we fixed and permeabilized them and also did the immunofluorescence staining. for the virus and for the receptor CD163. And for the supernatants, we did the titration. Now let's move to the result. Firstly, these pictures are the confocal images about the viral attachment. There are four columns totally. They are separately negative control. The three isolates 3249, 3262, 3267. And the first two rows is about the viral attachment on immature bone marrow-derived dendritic cells. The last two rows is on the mature bone marrow-derived dendritic cells. In this picture, the green color stands for the receptor CD163 or CD169 staining. The red one is for the virus, and the blue one is for the nuclear DNA. Now let's move to the first row. It is very obvious that except for the first row is about the combination staining of the virus and the receptor CD163. It's obvious that except for the CD163 positive cells, there are also CD163 negative cells could also be attached by the virus, and the red arrow is about the negative cells but with attachment. The same thing happens to the third row. It is the same staining combination, but on mature bone marrow dendritic cells. And the second row and the fourth row is about the combination staining of the CD169 and the virus on immature and mature bone marrow dendritic cells separately. It's the same that also CD169 positive and CD169 negative cells could both be attached by the virus, but here there is a special case about the isolate 3262. We can see the first two rows about this isolate. We couldn't detect the attachment on immature bone marrow-derived dendritic cells about this isolate, but however, we could detect the attachment on mature bone marrow-derived dendritic cells. Except the attachment, we also examined the replication of these three isolates in immature or mature bone marrow-derived dendritic cells. The pictures here are my negative controls at 3 different time points and the supernatant titration. As expected, there is no virus found here. Now let me show you the replication of the 3 isolates. The infection was examined at 3 different time points, 12, 24, and 48 hours, and in immature and mature bone marrow-derived dendritic cells separately. In the confocal macrophages on the left, the same. The green color stands for the receptor CD163. The red one is for the virus, and the blue one is for the nuclear DNA. We can see from these pictures that cells with CD163 labeling and the cells without CD163 labeling could both be infected by the virus, and the same case for the 3 different isolates. The graphs on the right is about our titration result of the supernatant. The dot line is the mature dendritic cells, and the solid one is the immature bone marrow-derived dendritic cells. It is very clear here that the replication efficiency in immature bone marrow dendritic cells is much higher than in mature bone marrow-derived dendritic cells, and the same case is also for the three different isolates. But it is worth noting there, the special isolate 3262, which one had no viral attachment on immature bone marrow-derived dendritic cells, however, had very good replication in immature bone marrow dendritic cells, even than in mature bone marrow dendritic cells. Another remarkable fact is why the replication efficiency in immature bone marrow dendritic cells is much higher than in mature bone marrow dendritic cells. For this, we did the flow cytometry. The flow cytometry to detect the CD163 expression level. The upper row is my relevant control about in immature and mature bone marrow-derived dendritic cells separately. It is very obvious that there is almost 15% more of the CD163 positive cells in mature dendritic cells than in immature dendritic cells, which is contradictory to our result that the immature bone marrow dendritic cells could better support the PRRS virus replication. That means other receptors beyond CD163 may exist. Except for the comparison of different isolates in immature and mature dendritic cells, we also compare the difference between each isolates. From these two graphs, we can see that the isolate 3267 replicated much faster than the isolate 3262, and they have statistically difference. Okay, from my study, we can get such kind of conclusions. 1. Siloadhesion and CD163 probably are not the only receptors for PRRS uses for binding on target cells. Second, from our results, it can be suggested that some CD163 negative bone marrow-derived dendritic cells support PRRS virus infection, replication. Third, activation status of bone marrow-derived dendritic cells is related to viral binding and virus replication. Fourth, the rate of CD163 expressing cells among bone marrow-derived dendritic cells is not proportionally correlated with virus replication.